going to present our project, Fusarium wilt, which focuses on the world's most famous yellow crescent-shaped fruit, which is, of course, the banana. And it's currently being threatened by fungus called Fusarium oxysporum, Forma specialis cubans, tropical race 4, which is kind of long, so we're going to shorten it down to FOC. Now, for a little background information, what we know as bananas are actually lots of groups of clones called cultivars. And one of these cultivars is the Cavendish, which makes up around 99% of all exported bananas to the US and the EU. And due to the lack of diversity in clones, these guys are being threatened by many diseases, and the most fundamental of which is FOC. And around 70 years ago, a similar disease wiped out the previously popular Gros Michel variety of bananas, and which the Cavendish then replaced because it resisted FOC race 1. But now the Cavendish is under threat from FOC tropical race 4. And FOC tropical race 4 has already spread throughout the world, from Asia to Australia to Africa, and more recently in 2021 to South America. And so because of this, we wanted to model all of the areas where FOC could cause the most damage. And we did that using MaxSet to find the most suitable areas for bananas. And the most suitable areas for bananas would be the areas at most risk of FOC. And so those suitable areas are in the dark blue here. And we found that these areas include countries such as Ecuador, Venezuela, Brazil, Indonesia, Malaysia, and more. Furthermore, with our model, we brought it to simulate the future, where we created a map of the future in around 40 years under simulations of climate change. And we found that banana-suitable areas increased by over 500 million hectares. And that indicates a larger banana economy and the more damage FOC can cause. And so, what does FOC actually do? Well, FOC is a soil-borne fungus that invades through the roots. And when it finds banana roots, it punctures through using gaps and wounds, and then travels through the vascular system, rapidly spreading and reproducing, causing the signature yellow wilt of the disease. And for all of this, the current solutions, such as quarantine fungicides and genetically engineered bananas, don't solve the fundamental problem with FOC, which stems from the lack of genetic diversity in bananas and their monoculture. And so this is where we present our solution, a fungicidal rhizobacteria that would defend the banana tree from harm. And our hope is that our solution will adequately defend the tree and stall the situation for long enough for us to develop a new, sustainable and more genetically diverse banana market in the future.
So the goal of our solution would be fundamentally to have local defense of banana roots and to have autonomous action against FOC and to have proper biocontainment due to the strict legislation surrounding GMO application to food. So in order to further investigate the actual details of our solution, we first analyzed the actual infection dynamic between FOC and banana roots. So essentially, how FOC invades into banana roots is through the detection of banana root exudates. And FOC chlamydospores basically take this root exudate as a signal to germinate and extend its fungal hyphae to infect the banana root. And the root exudate then also contains this compound called malic acid. And by the principle of diffusion, it then produces a malate sufficiency zone and a malate insufficiency zone. So we plan to take advantage of this sort of dichotomy of soil regions to engineer a bacteria that exclusively resides in this malic acid sufficiency zone, secreting chitinase in order to defend against the FOC through the destruction of its invasive hyphae. In order to actually control the FOC, to control our engineered bacteria to this malic acid sufficiency zone, we devised a toxin-antitoxin system that responds to the signal of malic acid in order to control its presence. So in the absence of malic acid, CCTB dominates and thus the bacteria dies. And in malic acid sufficiency areas, CCDA dominates and the bacterial death is not induced. So in order to do this, we found a malic acid responding transcriptional activator in order to govern our antitoxin. So here you can see that MLER is constantly transcribed by the T7 promoter, and it responds to L-malate to form an activated form of the transcriptional activator, which induces the expression of the antitoxin, allowing the bacteria to survive against the constantly and the constitutive and weakly express CCDB that will slowly kill the bacteria in the absence of the permissive environment of the malic acid. Then going back to devise the plan to actually inhibit FOC growth, we found in literature a novel chitinase called chi-18H8 that has been demonstrated in literature to have successfully inhibited FOC growth. We then subsequently linked it via GIS linker to a YBF tag in order to allow for extracellular secretion such that the secreted chitinase can actually interact with the cell wall of the hyphae to actually induce inhibition. And going back to the antitoxin model. So in order to model the complex behavior of all our constructs involved in the toxin-antitoxin system, we devised a kinetics-based model in order to basically estimate what malic acid concentration is required to keep the bacteria alive. Simultaneously, we also did a literature-based sort of exponential regression to estimate the amount of malic acid in that MA sufficiency zone that was mentioned earlier. And this value was roughly 25.3 micromolars. And comparing that to our results, we can see that CCDA dominates over CCDB at concentrations of malic acid around 1 to 10 micromolars. And therefore, this sort of preliminarily demonstrates that our toxin-antitoxin model has the potential to work. Then for our proof of concept, we essentially basically demonstrated that the chitinase was successfully exported out of the cell, and this has the potential to interact with FOC. To do this, we performed SDS page on the internal cell fraction and the extracellular supernatant. The results show that there are 46 kilodalton bands in both the intracellular fraction and the extracellular medium, and this demonstrates that chitinase has been secreted successfully out into the environment, and this allows for the potential inhibition of FOC. And we also did further SDS page in order to confirm the expression of all of our intended proteins in the toxin-antitoxin system. However, CCDA was not confirmed due to the fact that our buffer system, lamellae buffer system, is poor at resolving small peptide bands, so we could not confirm the expression of CCDA. And these are just gel pictures confirming the expression of CCDB at around 13 kilodaltons and chi-18H8 at 46 kilodaltons. So in order to further demonstrate that the secreted chitinase has the potential to inhibit fungal growth, we simulated bacterial antagonism against FOC using yeast instead. However, we were unable to collect any sort of conclusive results due to the fact that yeast propagates extremely slowly. This is a picture at roughly 2 weeks of growth, and as you can see, there's still a significant gap between the interacting parties. So basically, to improve this, we might shorten this gap, or instead basically contact bioresource agencies to source safer strains of Fusarium oxysporum to experiment with. Our future plans for experiments include further characterization of our toxin-antitoxin constructs to further fine-tune the system to allow for proper biocontainment, to step forward in the legislations that are limiting basically the application of GMOs in the field.
To understand the practicality of our solution, we approached experts who told us the many nuances of real-world implications of our project, specifically Dr. Andre Drenth of the University of Queensland, who has an extensive research background in horticulture, plant pathology, and forestry. Upon discussion, Dr. Drenth suggested implementation of our solution at earlier stages of plant life to overcome root turnover and provide sufficient bacterial coverage to the root system of the banana plant. Reflecting upon Dr. Drenth's feedback, our team plans to periodically apply our solution to ensure proper bacterial coverage, proper bacterial concentration for defense. To this end, we decided to deliver our bacteria through a liquid inoculant. Our visit to the Taiwan Banana Research Institute, a banana cultivation facility, allowed us to expand upon our knowledge on bananas and helped clarify Dr. Drenth's advice. By observing the plantlet production process firsthand, our team saw the possibility of applying our liquid inoculant to individual plantlet jars within the facility. To tackle the issue of the developing root system, we decided to integrate our solution to the already existing drip irrigation system through a Venturi pump, also known through the Taiwan Banana Research Institute. With an established implementation plan, we approached Dr. Li Yu Liu of the National Taiwan University, who told us about the many regulations regarding genetically modified organisms. This inspired us to look further into both international and national policies regarding our implementation method. Both these experts allowed us to create a more comprehensive entrepreneurship page and a more cohesive and applicable hardware plan.
Then, to estimate the total market loss caused by the facility wealth and the total amount of savings that our hardware can provide, we create two economic models. And this is done by combining fungal spread prediction, price prediction, and disease rate reduction prediction. The total market loss is estimated to be $50.3 million by the investigation of 30 years. However, under the full protection of our prototype, the economic loss is estimated to be reduced by $16.7 million, thereby significantly buffering the crisis. In an effort to gain the public opinion, our team has conducted a public opinion survey, which to our surprise showed a majority of respondents do not understand the ongoing crisis. Addressing the results of our survey, we plan to educate the public in 3 different approaches: presentations, podcasts, and the infographic translation project. Through in-person presentation, we engage with the audience with traditional teachings and detailed information. To create easily and digestible content for people who are interested in biotechnology, we created a podcast channel detailing the topics regarding the morals and the ethics about synthetic biology and the new advancements of this scientific community. Last, we create the infographic translation project where we partner with 3 other teams, and we hope to surpass the language barriers and spread the knowledge of synthetic biology through entertaining and understandable formats.
To the dilemma posed by the Fusarium world pandemic, our response is composed and clear. There will be, Fusarium won't. Thank you for listening.